Welcome back. In this series of video, we have already discussed about the different types of receptors which are basically present on the surface of cell. Now here we are going to discuss one of the important aspects of cell signaling, that is the GPCRs. So these are G protein coupled receptors. Now these are most important receptors and they perform a wide range of the functions. Now the G proteins, they are basically responsible for sight, they are responsible for smell, and they are responsible for the taste. Okay, now let's have a look at the GPCRs, how they actually look like. So this is a typical example of the receptor which is lying embedded in the surface of what? In the surface of the plasma membrane. Okay, so you can see this is an inactive GPCR because there is no signaling molecule till now which is bound to this receptor. Now you can see that this is the receptor and this is the G protein. Okay, so we have already discussed in the previous video that the G protein is basically made of three subunits, that is alpha, beta, and gamma. Now alpha and the gamma subunit, that is tethered to the surface of the plasma membrane. Now you can see here that since there is no signaling molecule which is bound to the receptor, so the GPCR, that is in an inactive conformation, and when it is inactive conformation, it is bound to the molecule of GDP, that is the guanosine diphosphate. Now consider here, once a signaling molecule comes and it binds to this receptor, what happens is that, so the work, basically the signaling molecule and the binding of the signaling molecule to this receptor, it is similar to what a guanine exchange factor does. Okay, so we see here an inactive form has a GDP bound, and once the signaling molecule binds, it changes the conformation. So from the inactive conformation, it's now in an active conformation. And once activated, it leads to the activation of G protein. So you can see that here the molecule which is bound here, it is GTP. So basically this binding of signaling molecule to receptor, it has acted in the same way as a guanine exchange factor acts. So here has been the substitution or a replacement of the GDP, and a GTP is placed in that position. Okay, so both our alpha subunit, that is in an active state, and our beta subunit, or rather we can say it is the beta and gamma complex, that is basically what, that is now active. Now it's not only that alpha subunit, it will activate other proteins or it will have an effect on the cell. Both of them, they have the effectors, they have their effectors, both alpha as well as the beta and gamma complex, and they lead to the different changes in the cell. Now let's again picture the same GPCRs but in a much more easier way. So you see here we have suppose a hormone is coming, so that is basically what a signaling molecule is. Now signaling molecule, a hormone comes and it binds to the surface of receptor. Okay, so here is the binding of this. Now once this binding basically takes place, so consider this is the same serpentine receptor we have been discussing earlier. This is the G protein coupled receptor. Okay, so this receptor is basically bound to this G protein, which is a three subunit. So this is alpha, beta, and gamma. Okay. This binding of this hormone to its receptor, it leads to activation of what? Activation of the G protein. So this is activated G protein, and once this activated G protein, it now again what it does is it activates another enzyme. You can see that it's again bound. We can say that it traverses the plasma membrane, similar to that of the receptor GPCRs. So this is the adenylyl cyclase. What basically its function is that it catalyzes the formation of cyclic adenosine monophosphate. Now that's very important. So this enzyme basically catalyzes the formation of CAM from the ATP. Using ATP, it does the formation of the CAM. Now basically CAM, this is a very important secondary messenger. So we already discussed that we have a lot of secondary messengers and they perform, most important we can say, they perform various functions in our body. Okay, so this CAM we can we'll ahead in our next slide we'll see briefly that what basically different types of the functions CAM performs. Now let's move ahead and discuss another important thing. So the same thing which we discussed previous, this is the inactive state because there was no, so you can't see that there's no binding between the signaling molecule or suppose a hormone or a receptor. So this is in the inactive state, a GDP is bound, so this is fine. Now once a receptor basically, or let's suppose that once a hormone binds to this receptor, so that means an activation is taking place. This is once this is active, you'll see that in place of GDP, now we have a molecule of GTP bound to the alpha subunit. Okay. Now, the same thing which we saw in the previous slide, the alpha subunit and the beta gamma complex, they will have their target enzymes or maybe ion channels. So they will effectively bind to their effector protein. So they will have their own function. Now, the most important thing which I want to mention through this cycle is that, so we think that only the activation is important. So it's not like that now once this is activated, as much as we say that the process of activation is important, same is the case with the off state. Okay, so once you have activated basically a receptor, you need that this receptor, it should be again now in an off conformation, so it should be switched off because you need this receptor to function again. Okay, so the same thing happens here. Now consider that now this was in an active conformation. Now what happens is that, so if it has to turn from an inactive conformation to an from an active conformation, sorry, to an inactive conformation, now what it will do is that it will hydrolyze this GTP. Now once hydrolysis of the GTP occurs, GTP triphosphate, it will be hydrolyzed, and after hydrolysis again you will see that it will again go into the inactive conformation and GDP is bound here. Okay, so that is basically, we can say that, so the both the switching on and switching off of the signaling molecules, it's equally important. Okay. Okay. So we talked about the molecule which said that this is a cyclic adenosine monophosphate which we have generated till now. We discussed about it. This is a very important secondary messenger. Okay. Now the secondary messenger, it comes and it binds to another protein. That is our protein kinase. So you see here, this is the representation of the protein kinase. So this is basically protein kinase A. Now, remember this thing that protein kinase is basically made up of 4 subunits. 2 of them are regulatory subunits and 2 of them, which are shown in the blue, these are catalytic subunits. Okay, so they'll have the catalytic function. These are the regulatory subunits. Now, from the picture, it's very clear that cyclic AMP, which are generated from the signaling of the GPCRs, they'll bind to these 2 receptors. So these are the regulatory subunits. They will bind to it. Now, once this binding occurs, it will lead to the release of the catalytic subunits and they will be now active. Okay, so once the catalytic subunit, the protein kinase, is active, you can see that these active catalytic subunits, they will have their own set of proteins, own set of effective proteins to bind to, and they will lead to activation of these proteins. Okay, now consider here another important thing about the GPCRs. So see here, so we have a signaling molecule, and let's have a quick recap till now whatever we have discussed. So we have a signaling molecule, it binds to this receptor. So our receptor, it's activated now. You can see that we have a GPCR and a GTP molecule is bound to it. This represents the activated adenylyl cyclase, and it leads to the production of the cyclic AMP from the ATP. So you see here ATP, and the formation of the red dots represents the CAMP. Now once the CAMP is produced, it will bind to the regulatory subunits, which leads to the activation and release of the protein kinase A. This same protein kinase A, what it does is that, as I said, that it may have different kinds of the proteins which it can bind to and it can subsequently activate them. Similarly, what happens is that, um, so one of the proteins it basically binds is the CREB. Okay, so that is the, we say this is basically cyclic AMP response element binding protein. Okay, so once it basically binds to this receptor, it leads to its activation. Okay, the activated, or we can say it leads to its activation, this activated phosphorylated CREB, it basically activates the gene transcription, and this gene, it's basically leading to the production of an hormone, that is the somatostatin. So you can see how a signaling molecule led to a change in the nucleus, which eventually led to a change in the gene expression of a particular hormone. And we know that somatostatin, it has got many roles to play in brain and other functions of the body. So in the next, we'll again talk about another very important signaling molecule.